trial, successful or not, it tightens that target zone for the next one.
But okay, moving from prevention with a vaccine to an actual cure,
Mm.
that takes us into some really complex territory.
It does. We have to talk about latency.
Latency, right. This is the great scientific hurdle. For anyone who follows this, the virus sets up these reservoirs, hidden pockets of infected cells, almost right after infection.
And the current antiretroviral treatments, they suppress the active virus, but they can't touch these dormant copies of viral DNA that are integrated into our own cells.
It's the ultimate hiding strategy.
It is. And to tackle those reservoirs, the research is now organized around three really distinct and powerful pillars, often designed to be used together.
Okay, let's break them down. Pillar 1, gene editing. When we say CRISPR, we're talking about something with truly transformative potential, a permanent fix.
That's the idea. Gene editing offers sort of a dual strategy here.
Okay.
The first is disrupting the host pathways. A great example is targeting the CCR5 co-receptor.
And CCR5 is famous because of the Berlin patient, the first person ever cured. He got a bone marrow transplant from a donor who had a natural CCR5 mutation.
Exactly. CCR5 is basically the main doorway that most HIV strains use to get into immune cells. So if you can use a CRISPR-based approach to edit T cells and just remove that receptor,
You create cells that are inherently resistant to the virus.
you do. It's essentially a functional cure for those specific cells.
But the bigger goal, the one that really means eradication, is using CRISPR to go after the virus that's already hidden inside the cell. Is that right?
Yes, that is the huge aspiration. Using gene editing tools to literally excise, to cut out the integrated proviral DNA from the host cell's genome.
Wow.
If you can do that precisely in enough cells, you could theoretically eliminate the entire reservoir. It's still early days, but that possibility of a surgical permanent eradication is why this is such an essential pillar of the research.
So if CRISPR is the surgical strike,
Mm-hmm.
then pillar 2, broadly neutralizing antibodies,
Mm-hmm.
or bNAbs, that feels more like an active defense strategy.
That's a good way to think about it.
So what makes bNAbs so critical to this cure roadmap?
Well, bNAbs are these naturally occurring antibodies. They were isolated from a few rare individuals who have this incredible ability to neutralize a vast spectrum of HIV variants.
And the trials have shown they work for prevention and treatment.
Yes. For prevention, they can act as a kind of long-acting passive shield. But for treatment, they're really interesting.
How do they work toward remission?
If you administer high doses or combinations of different bNAbs, you can temporarily suppress the viral load in patients. They're being looked at as a key component in long-term remission strategies.
So they could let people stop taking daily pills.
Potentially. They could provide a bridge, control the virus while other strategies like latency reversal get deployed.
Which brings us right to pillar 3, the most direct attack on latency itself, using what are called latency-reversing agents, or LRAs.
Right. LRAs are basically the chemical keys designed to unlock those viral reservoirs.
The sources mention a few classes being tested: histone dehydrolase inhibitors, protein kinase C agonists.
They're mouthfuls for sure, but the fundamental biological process they're trying to disrupt is gene expression.
Okay, yeah.
So let's take the histone dehydrolase inhibitors. When the viral DNA is integrated into our genome, the cell often keeps that region bundled up really tightly. It's a process called chromatin modification, and it silences the viral genes.
It puts them to sleep.
Exactly. And HDAC inhibitors interfere with that. They chemically loosen that chromatin structure, which allows our own cell machinery to access and transcribe the viral DNA.
So it forces the virus to express itself. It makes it visible.
It forces it out of hiding. And once the virus is visible, researchers have two major and kind of contrasting strategies.
The first one sounds pretty dramatic: shock and kill.
The name is very precise. The LRA provides the shock, forcing the virus out of latency. Then, once those infected cells start expressing viral proteins, they become vulnerable to destruction.
That's the kill phase.
That's the kill phase.
And that involves more than just standard antiretrovirals, I assume.